Now we're going to talk about how important it is to accurately diagnose a cancer, or to take a cancer and a type of cancer and determine the subtypes within that type of cancer. And through accurate diagnosis, it's thought that we'll get better treatment. So new therapies will depend upon the accurate diagnosis of cancers. Now, by way of example, we'll discuss breast cancer and some of the outcomes from treatment. So traditionally, ways of classifying cancer don't tell you about the molecular detail of what's going on. Okay, if you're looking at the cancer down a microscope, or by the type of lump it is, or by some sort of way of looking at the cancer, it sometimes makes it more. If it's better diagnosed, you can treat it better. Okay, so often a single type of human cancer is not actually a single disease. It's actually a bunch of different diseases that come up with a similar appearance in the similar organ. Okay, so often we can have distinct subtypes of a particular type of cancer. Now, the reason this is important is that when you treat cancers, often the compound that's used to treat the cancer has strong side effects. And these side effects often are not just temporary side effects that you get over, but these side effects have a lasting detrimental effect on the body. And often the incidence of cancers at a second site are higher if you've been treated with one of these anti-cancer drugs. So if you think about cisplatin that we talked about earlier, cisplatin binds to DNA and causes all sorts of problems for DNA metabolism. This happens in normal cells and in cancer cells. Now, cancer cells are more susceptible because they're growing faster, but normal cells are also exposed to this drug that's damaging DNA and causing mutations elsewhere in the body, leading to second site cancers. Okay, so you don't want to use these drugs unless you absolutely have to. Another example, or a more specific example, would be from the textbook, would be looking at in the 1980s, looking at breast cancer patients. So breast cancer back then wasn't so much subtyped into these subcategories of breast cancer. It was seen under this big umbrella category of breast cancer. Okay, and we know there were subtypes. So then breast cancer generally was treated with a compound, cyclophosphamide. Okay, now cyclophosphamide is an alkylating agent, meaning it binds to and covalently attaches these alkyl groups to proteins, and I don't know exactly how it works, but DNA, and causes all sorts of damage. Okay, now if you are treated with this compound, you have approximately a 7-fold increased risk of subsequently developing leukemia. Okay, so the drug itself that's used to treat the breast cancer puts you in a higher risk category for another cancer, right? But if you've got a breast cancer that's going to have a high impact on your life, then taking the drug and having an increased risk for something else is a win. Okay, but if you have a breast cancer that's not going to cause you much problem, it's benign, then having this treatment has put you in a risk category when you didn't need to be treated in the first place. So it's a difficult balancing act, and it'll be easier to diagnose and treat once you understand these subtypes. So an accurate diagnosis of these cancers will then lead to a reduction of treatment when treatment wasn't necessary. Okay, so on the plus side, if you've got a fairly large cancer in the tissue here, treatment can significantly reduce it and therefore reduce the risks. But if this cancer was going to metastasize and spread around the body, then it's good that it's treated. If this cancer is going to be benign and not actually cause further problems, then does it need to be treated, particularly if the treatment is going to cause downstream effects, so if the treatment is going to make you more susceptible to other cancers. So, so for instance, if you can identify which breast cancers are more likely to become metastatic, then these are the ones that you need to treat. And if your cancer is going to remain benign, then it's probably not as necessary to treat these ones. Okay, so the majority of primary breast cancers in the United States have been treated aggressively with these drugs that and a fairly 15% of these 15% of these cases, maybe they were benign and were not going to become metastatic. Okay, so the upshot of this is we have to have better ways of diagnosing subtypes and then identifying whether that cancer, firstly, is going to be malignant or not, and secondly, can we predict whether that subtype will be responsive to treatment. So what we want is accurate diagnosis of cancer, and this has been happening recently with the combination of modern genetic techniques such as microarray techniques to look at gene expression. These are often referred to as gene expression arrays. So it's basically a microarray experiment. And you combine the experiment with some fairly complicated statistical analysis of the results, and then you can categorize a patient based on the gene clustering or whatever it is you're doing of the results, the clustering of the results. And with the combination of the modern genetics and modern analysis, you can predict the clinical course of breast cancer now with 90% accuracy. So if you can predict this is going to be metastatic, you treat it. And if you can predict it's going to be benign, then maybe you wait on treating it. Okay? And this now holds the promise of sparing many women from exposure to unnecessary chemotherapy. So, what is a gene expression array? So what is this modern molecular biology tool that we're using to diagnose a cancer into a particular subcategory, a particular subtype? Well, this is just showing a generic gene expression array experiment. Okay, so this slide here has lots of small dots on it, and it's called a microarray. So it's a very small array of, it's an array of lots of small samples of DNA. And each sample of DNA on this microarray represents a gene. Okay, so let's assume that for every gene in the body, there's one spot on the array. Okay, so I know that that spot there, for argument's sake, is RAS. And I know that that spot there is, tumor suppressor protein 53, and I know that, I know the identity of all of these spots, okay, because they've been spotted by a machine, and we've spotted particular gene sequences in particular spaces. So we know where they are. Okay, so we're looking at the all genes in a cell in one experiment. Now, this slide is commercially bought and is used for the experiment. Now, at the top here is that we have the normal cells and the cancer cells. So these are our patient samples. Okay, so we take from normal cells, we look at what genes are being expressed. In other words, we look at the messenger RNA. Sorry, we look at messenger RNA levels in the normal cells. So we isolate the messenger RNA, and the messenger RNA tells you that a gene's being expressed. Approximately, it tells you that. So we look at the messenger RNA level. We convert the messenger RNA into an equivalent molecule called a cDNA, and it's labeled so we can detect it. So the messenger RNA is converted into a detectable signal, and because this is single-stranded, and because these DNA spots are single-stranded, then the labeled message here will bind to the gene it was expressed from. So if I've got levels of p53 being expressed, I'll have levels of p53 messenger RNA. When I convert that messenger RNA into a green fluorescent label, that messenger RNA will bind to the particular gene, and it won't bind to the other genes. Do the same experiment in cancer cells, extract and isolate all of the messenger RNAs that are being expressed, convert that to a labeled cDNA that we can detect, and then we hybridize the single-stranded DNA to the slide, and it will find the gene it was expressed from, and it will bind to it. Okay, complementary DNA base pairing between complementary strands. So what you then have is, if a gene is colored red on this slide, then it means there was more of it in the cancer cell than in the normal cell. Okay? If a spot on this slide is colored green, it means that there were more of that gene expressed in normal cells than in cancer cells. And if it's orange, then it was expressed equally, because red and green make orange. Okay? So by the color of each gene in the genome that's on this slide, you can tell whether that particular gene was on or off in a cancer cell compared to a normal cell. So it tells you in exquisite detail what the level of gene expression is. So are your tumor suppressor proteins on or off? Are your oncogenes being overexpressed? Are genes involved in cell cycle regulation up or down? If you know which players are up and which ones are down in a particular cancer, then you can target what's on. You can target, ooh, we know this gene's on, that's going to make the cell grow, let's target it. Okay, just to read off this list here, to try and, do what I've just expressed to you. The core principle behind a microarray is the hybridization of two strands of DNA. Okay, so we have the strand derived from a messenger RNA telling you the gene was on, hybridizing to the spot that is on the slide. And all of these single-stranded sequences find their partners on the slide, and then once you remove all the non-specific binding, then the fluorescence color will tell you whether the gene was on in cancer cells relative to normal, because it would be red, or whether the gene is more strongly expressed in normal cells, in which case it will be green. Okay, so you can look at relative gene expression between your sample and your normal sample. So this is the result of a microarray experiment. So you take this slide and you measure all of the intensities and you put it into an Excel spreadsheet. And rather than looking at numbers, we're looking at colors. Okay, and if the number was indicating it was underexpressed in the cancer cell, then it's colored green. If the number suggests that it is overexpressed in a cancer cell, it's colored red. And then what we have here, for each sample that was looked at, so for each tumor sample we looked at, so you take the top row here, the very top row, then from patient sample number 295, this patient here, then they've looked at 70 genes. So the row is the patient and the columns are one of 70 genes. And by looking at the color, you can look at whether that gene was overexpressed here or underexpressed. And then you use your bioinformatics to group all the genes that behave similarly. And then you can then say that, for instance, the a poor signature will have this kind of profile. So there's a whole bunch of genes that are red, which are overexpressed in the cancer cell. And if these genes are expressed in this tumor, this tumor, this tumor, and this tumor, we know from looking at the patient history that expression of these genes correlates with a poor outcome. So 75 out of 80 patients will undergo metastasis. So that's a sort of a very dangerous cancer. Okay, if your patient sample shows, rather than these genes being highly expressed, but these genes being underexpressed, and there's another set of genes here that are overexpressed in the cancer, if you have that profile, then that's a good indicator, because only a small proportion of these patients undergo metastasis. So about, 15%, as opposed to about 50%. So by looking at what genes are on and what genes are off for a particular sample, you can then use that as a predictor of the severity of that cancer. So now we have techniques where you take patient samples, you can do the analysis, and you can see whether it's a predictor of a highly malignant cancer or a more benign cancer. And therefore you can make the decision of whether this cancer needs to be treated or not. Okay, there's another really good example in the literature or in the textbook, depending on how you want to look at it, of how you can take what was considered to be one type of cancer that was difficult to treat, and to break that one type of cancer into 3 subtypes. And then you can identify one subtype that is responsive to treatment and one subtype that's not responsive to treatment. So rather than having, well, I've got this cancer type and I may or may not be responsive to the drug, you can say, well, I've got this subtype of the cancer and I know I'm going to be responsive to the drug, or I know I'm not going to be responsive to the drug. And that's a much more powerful way of diagnosis and treatment of a cancer. So accurate subtyping of these cancers. So let's quickly have a look at B-cell lymphomas. So they've been difficult to treat in the clinic because it seems to be such a varied response to the drugs. They've used these gene expression arrays that I've tried to explain to you in a bit of detail. You don't need to know all the ins and outs of how a microarray experiment works, but I've explained to you how they work in a fairly simple way. So you can use these gene expression arrays to try and segregate the disease, the single disease, into subtypes. So this is what B-cell lymphomas look like when you take a patient sample and you put it on a slide. Now, this disease is difficult to treat. But if you then do microarray analyses on these samples, you can split it into one of 3 different subtypes. So we can now say that a patient sample isn't just B-cell lymphoma, it's a primary B-cell lymphoma. Or it's one of these ones, it's a germinal-centered B-cell lymphoma. Or it's an activated B-cell lymphoma here. So you do the microarray experiment, and then you can subtype this sample into this, this, or this. All right? And then it turns out that you can study the molecular biology of what's going on in these 3 different types, and you can use that to inform the treatment. So here we have the 3 types. Here we have the results of microarray experiments. And here we have some representative genes. So for the primary B-cell lymphomas, then there's a bunch of genes here that are highly expressed in these ones. Okay, so let me start again. We've taken these cells and we've looked at the expression levels. If the expression levels are high, then and they're low here, then you can say, well, in this cancer type, we know these genes have been perturbed. Whereas if you have this cancer type, then it's the opposite. The genes down here have been perturbed strongly, where these ones have been perturbed less. So these genes are turned on, these genes are turned off in these different subtypes. So without going into too much of the details here, it just tells you that you can identify which genes or which gene targets are on and which ones are off. And if you look at these genes that are on and off in these different subtypes, you might say, well, I'm not an expert. I can't see anything there that helps me. But what it turns out that if you look at the control of expression of some of these genes, some of them have similar control of expression. So if you target the transcription factors that are turning the genes on, then you can fight the disease. Okay? So even though the genes are diverse, they have similar control mechanisms of activation, of turning the gene on or turning the gene off. So you can target that. So when you look at these three categories that have been identified through looking at gene expression arrays, the activated and primary categories have high levels of NF-kappa beta activity, which is a transcription factor which is turning on a whole bunch of genes. Okay, so if you can target NF-kappa beta, then you basically can turn off all of these genes and fight the disease. Whereas the other category that has been identified here, these germinal cells, go back to, I can't remember what they're called, these germinal cancer centered B-cell lymphomas, that category are independent of the NF-kappa beta transcription factor, and therefore these will not be sensitive to the treatment that will work for these. So by subtyping, you can identify this category will be responsive and this category won't. So just looking at the survival rates of these categories, if you look at the patient outcomes in relative to the microarray results, so that we know we now have three categories, there's actually distinct patient outcomes for the microarray categories, indicating that the microarray categories are a very real distinguisher of the subtypes. Okay, so if you have the primary one, then you can say that your chances of survival are are higher than if you have another category. So you can predict the severity of the disease based on the microarray profile, and more importantly, or advantageously, let me just jump a slide here, you can say that because we know NF-kappa beta is the transcription factor driving gene expression, you can use an inhibitor, I-kappa beta, an inhibitor of the NF-kappa beta, and if you inhibit that transcription factor, which is the case for these two, they're the ones where the NF-kappa beta has a strong role, then the viability of those cells decreases. Okay, so you're actually treating the cause of the cancer through a specific inhibitor for a particular molecule. And for the subtype where NF-kappa beta is not involved, using the inhibitor of the kappa beta has effectively no effect. So here we're now starting to have optimism that if we understand the subtype and what's causing it, we can treat it. And if we know that you don't have that subtype, then the treatment's not going to work, so you wouldn't treat it. And when we talked earlier about cancers, we talked about multi-hit hypothesis, and different players were involved. Like different cancers had, it might have RAS and it might have P53 and it might not have this, and different players were involved. And if you look at these three categories, then for the primary ones, then there's a fairly good percentage that you're going to have these genes being involved. Whereas if you have another category, the different subtype, the activated one, then these genes are not involved in that particular subtype of the cancer. So even though they look the same under the microscope, there's different factors driving the cancers. And therefore, you need to treat them differently in the clinic with different drugs. So you can think about them as separate diseases. And yeah, so there's a few other things, places in the chromosome where there are genes that haven't been categorized that are not involved in some subtypes and are involved in some of the other subtypes. Okay, so accurate diagnosis leads to better predictability of outcomes in terms of malignancy and benign, and also gives you a better chance of designing a drug for that cohort. And just lastly, if you were to treat all of these cancers as a single disease, then the response to the drug treatment would be a low survival. You wouldn't have much of an impact because you've got some subtypes being non-responsive and some subtypes being responsive. Whereas if you just look at the response of the subtypes to the drug, then you'll see a much stronger positive result of this cancer subtype was responsive to the drug. And that's really important when you're trying to assay to see whether this drug is effective against this cancer. If you might be really effective at 10%, but 90% is not responsive, so the drug looks like it doesn't work. But if you were to just treat those 10% that are responsive, you'd have 100% efficiency. So you need accurate diagnosis so that you can accurately assess the effect of the drug. Okay, so we'll go on now to look at some successful drug treatments.